Good morning, everyone. We're going to get started. Today, we're going to be talking about estimating leaf traits from optical properties across a latitudinal gradient in Arctic Alaska. Today, we'll be hosting Ian Shuman from the Earth and Environmental Sciences Department at Columbia University. This research is a product of our research partnership program. So we lent Ian an instrument, and he was able to do some great research in his field work in Arctic Alaska this last summer. So he'll tell you more about it. Just to start out, I'm going to introduce Spectral Evolution. Spectral Evolution is the leading manufacturer of high-quality UV-Vis, NIR spectrometers and spectroradiometers. We were established in 2004, and we're located in Massachusetts, just outside the Boston area. Our primary customers are in hyperspectral applications, remote sensing and vegetation, mining and geology, as well as photonics and calibration transfer. So our instruments have uses in research as well as in industry in all of these different subjects. To speak a little bit more about Ian, who will be talking today, he is a second-year PhD student in Earth and Environmental Sciences at Columbia University. As part of Natalie Bowman's lab at Columbia, Ian's research combines field ecology and remote sensing to investigate the impact of climate change on terrestrial Arctic ecosystems. In particular, Ian is interested in how climate-driven changes in vegetation produce to change in plant physiology, the surface energy budget, and songbird habitat across the Arctic landscape. Ian is also part of the evolving Meta Ecosystem Institute and NSF Biology Integration Institute, where he is working with a diverse set of wildlife ecologists, geochemists, geneticists, and modelers to understand the eco-evolutionary responses of Arctic organisms to continue warming at high latitudes. So with that introduction, I'm going to let Ian take it away, speak a little bit more about his department and his research that he conducted this year.
Awesome. Thank you so much, Mackenzie. See if I can share my screen. Does that look right for everyone?
That looks great.
Awesome. Yeah, my name is Ian Shuman. I'm a PhD student at Columbia University, and today I'll talk to you guys about how I'm estimating leaf traits from optical properties across a latitudinal gradient in Arctic Alaska. So I'm sure nobody in this webinar needs any sort of introduction to climate change, but something you all may not be as familiar with is the fact that the Arctic is warming 4 times faster than any other ecosystem on the planet. This process, which is known as Arctic amplification, has resulted in nearly 2 degrees Celsius of warming in high latitudes already, which is something that we can see in this temperature anomaly product put out by NASA GISS. This warming, in turn, has resulted in massive changes to Arctic ecosystems, mainly by changing wildfire regimes, permafrost dynamics, nutrient cycling, and vegetation composition and species distributions. Of these many different changes, my collaborators and I and the rest of our research group, as part of this Spectral Evolution research partnership, are particularly interested in a process known as shrub expansion, which is one of the strongest hallmarks of climate change in Arctic terrestrial ecosystems. Shrub expansion, or just shrubification for short, refers to the rapid expansion in range, height, and density of deciduous tall woody shrubs across the Arctic. And these deciduous tall woody shrubs are primarily things like birches, willows, and alders. And we can see just how extreme this process of shrubification is by looking at these 2 satellite images of the Siberian tundra. The top one, taken in 1966, shows some shrubs across the landscape, but also plenty of open shrub-free tundra habitat. In the bottom picture, taken in 2009, we see that all of this open tundra has been filled in by shrubs. Shrub expansion is also widespread across the Arctic. So in this figure on the right, we can see how shrubification has contributed to a spectral greening trend across the Arctic, which means that there is more and more productive vegetation anywhere we see green colors in this map. And we see those green colors in Alaska, Canada, Greenland, Scandinavia, and Siberia. As a consequence, shrub expansion has had widespread effects on ecosystem functioning and is often thought to be a positive feedback to further Arctic warming. Furthermore, shrubs differ from other Arctic vegetation in both their physiology and their functional traits. So as shrubs expand and become more prevalent across the Arctic, these differences in traits can ultimately affect how terrestrial Arctic ecosystems function and how they respond to future climate change. For those of you who may not be as familiar with plant biology, however, what does it mean for these shrubs to have different functional traits and physiology? For starters, functional traits describe a plant's growth strategy. And for functional traits, we typically think of plants as falling somewhere along a leaf economic spectrum, where one end of the spectrum describes early successional plants with access to lots of resources that grow and photosynthesize very rapidly, but typically don't live as long. So this is almost a live fast, die young growth strategy. An example of a plant that falls on this left end of the leaf economic spectrum is something like maybe a roadside weed, but in the Arctic, we typically think of those shrubs that are expanding, those willows, alders, and birches, as falling on this side of the leaf economic spectrum. On the other hand, we have plants that are adapted to withstand environments with low resources. These are typically late successional species that are slow growing and longer lived. So in the Arctic, we could think of something like a spruce tree or an evergreen shrub as being on this end of the spectrum. This leaf economic spectrum is typically defined using functional traits. So the functional traits of a plant establish where it falls on this spectrum. One of these traits is leaf mass per area, or LMA, which just describes how thick a plant's leaves are. With plants on the left end of the spectrum typically having very thin, cheap leaves that are very productive but don't last very long, whereas plants on the other end of the spectrum have thick leaves that aren't as productive but last a long time. And so a spruce is a good example of this because we know spruce needles are very thick and waxy and they last many seasons. Similarly, the leaf economic spectrum is also defined by foliar nitrogen, which describes how much nitrogen is in a plant's leaves, either by mass or by area. Slow-growing individuals typically have low foliar nitrogen, whereas the fast-growing individuals on the left end of the spectrum have lots of nitrogen in their leaves because they're allocating this nitrogen to photosynthetic machinery that allows them to grow a lot and live fast and die young. In addition to functional traits, vegetation can also differ in its physiological traits, which describe how a plant takes up and uses resources like carbon, light, water, and nutrients. So an example of a physiological trait that we're interested in is something called stomatal conductance, which describes how open a plant's pores or stomata are to allow gases to exchange in and out of the leaf, with primarily carbon dioxide coming into the leaf for photosynthesis and water leaving through the plant's pores via transpiration. Similarly, another physiological trait that we're interested in is VC-max, which describes the maximum rate at which the enzyme Rubisco can fix carbon dioxide by combining it with another compound to create organic carbon like carbohydrates. If you're not familiar with plant biochemistry, just think of VC-max as being the maximum rate at which a plant's leaves can capture carbon dioxide to use it for photosynthesis. VC-max is a really important physiological trait as it's a key parameter in terrestrial biosphere models, as it's used to establish vegetation's photosynthetic rate, which in turn determines how large of a carbon sink terrestrial vegetation is. Ultimately, both functional and physiological traits describe how well a plant grows and where its nutrients are. And these leaf traits can differ both between and within different species of plants. So a process like shrubification may alter leaf traits by virtue of changing the species composition of tundra, as a greater proportion of plants will be shrubs and thus have thin, cheap leaves that photosynthesize a lot. However, the shrubs that are expanding in one part of the Arctic may have thicker leaves or may photosynthesize less than the shrubs that are expanding in another part of the Arctic. And both types of variation in leaf traits arising from differences between species and among individuals within a species can affect the functioning and resilience of the Arctic ecosystem. As such, our research group is interested in understanding the following two questions. What are the spatial and environmental controls of leaf traits across the Arctic? And how do leaf traits vary both between and within species? In trying to answer these questions, however, we have kind of a big problem, and that's that leaf traits are really hard to measure, especially in the Arctic. Getting data on the spatial and environmental controls of leaf traits requires hauling lots of heavy equipment to remote, hard-to-reach field sites. Weather in the Arctic is often uncooperative, and the Arctic has very limited infrastructure. Furthermore, a lot of these leaf traits just take a long time to measure in the field. So that VC max parameter that I mentioned a couple slides ago can take 45 minutes to measure for a single individual. And we often need multiple replicates of many different individuals in order to characterize this leaf trait at a given site. And so that long measurement time gives us time in the field to do things like pick blueberries and take tundra naps, but it's really not the most efficient use of our resources while in the field. This is where our research partnership with Spectral Evolution comes in, because many of those leaf traits that I just introduced, both functional and physiological, are ultimately determined by leaf biochemistry. Leaf mass per area, for example, is affected by the thickness of plant cell walls, and VC max is determined by temperature and the prevalence of enzymes in the leaves. In addition to affecting leaf traits, leaf biochemistry also affects leaf reflectance. So on the right here, I'm showing the spectral reflectance curve of vegetation, which I'm sure many of you are familiar with. And as you'll know, different regions of this spectral reflectance curve are influenced by different chemical compounds in leaves. So on the left visible end of the spectrum, we see absorptions from leaf pigments like chlorophyll. And on the right end, we see the effects of things like water, protein, lignin, cellulose, and other compounds. And so the theory goes, if leaf biochemistry affects both leaf traits and leaf reflectance, it should be possible to predict leaf traits from their leaf reflectance. And measuring leaf reflectance is significantly easier and faster than measuring leaf traits directly. So if VC max takes 45 minutes to measure in the field, measuring the reflectance of the same leaf can take less than a minute. And just to run through a couple examples really quick, one of those functional traits that I mentioned earlier was the amount of nitrogen in a leaf. And obviously, nitrogen is an element, and it's used in biochemical compounds. So those compounds have particular absorptions at different bands, at different wavelengths. And so we can use differences in the reflectances of those wavelength bands to predict the amount of nitrogen in a leaf. Similarly, about 30% of all leaf nitrogen is used to create photosynthetic machinery, which in turn determines the rate of carbon assimilation and ultimately VC max. That photosynthetic machinery has its own set of absorption bands, which I'm now showing here. And so it's possible to indirectly predict VC max from leaf reflectance via this shared photosynthetic machinery. This prediction is a little bit more abstract because VC max is itself an enzymatic rate or a physiological rate and not a physical structure itself. But because it's influenced by photosynthetic machinery, we can use the reflectance of that machinery to predict this rate. And so we can accurately predict both functional and physiological traits from quick and easy spectral reflectance measurements. And that's why this research partnership has been such a game changer for our team. But what exactly are we doing in the Arctic to characterize these leaf traits? For starters, we conducted a long field campaign in the Arctic last summer from June 12th to July 4th. And we took measurements at 15 different sites that were spanning a 350-kilometer latitudinal gradient. But before I get too ahead of myself, I'll first walk you through what we measured at each one of our sites, and then I'll walk you through where we did it and why. And so our order of operations in the field was this. First, we would measure the physiological traits of interest on plants while they were growing at our sites in the Arctic. And so here's a picture of me using a different instrument to measure VC max on a willow shrub at one of our sites. After measuring the physiology of the leaf, We would then measure the reflectance of the fresh leaf using a spectrometer from Spectral Evolution. We did this either while the leaf was still attached to the individual in the field, or we did it within 24 hours of clipping the fresh leaf from the individual in the laboratory. After measuring the fresh reflectance, we would then dry the leaves and measure some of their functional traits, so things like the mass of the leaf, its area, its nitrogen content, etc. And then after measuring the functional traits, we would measure the dry reflectance to remove the effects of water absorption from those reflectance curves. Most of this step was done in the laboratory, so here's another picture of me still measuring fresh leaves, but hiding in a tent from mosquitoes. And then finally, once we measured all of our different physiological and functional traits and fresh and dry spectra, we paired the leaf traits with the leaf's reflectance curves, and we used 80% of this data to train a partial least squared regression model to predict a given leaf trait from its spectra. We then used the remaining 20% of the data to validate the model's predictions. In case you're not familiar, a partial least squared regression, or PLSR model, is just a statistical model that's designed to handle large numbers of correlated predictor variables. And this is ideal for our application because, again, we're trying to predict a single leaf trait from 4,855 reflectance values. And obviously, there's some autocorrelation between nearby wavelengths. Partial least squared regression works by projecting highly correlated wavelength reflectance variables to a matrix with just a few latent variables. And it then maximizes the correlation between our response variable, our leaf trait, and those latent variables. As I said before, all of these measurements were taken between June 12th and July 4th of this past summer, and they were primarily taken at 15 sites spanning a large latitudinal gradient, 343 kilometers on the north slope of Alaska, which you can see in this red box here. We chose sites covering such a large spatial area as a space-for-time proxy to look at how these traits might change with warming, under the idea that the leaf traits at our southern sites, at the southern end of our latitudinal gradient, would resemble the future of the sites in the northern end of our latitudinal gradient under future Arctic warming. And at each of these 15 sites, we measured simple functional and physiological traits like leaf mass per area, foliar nitrogen, and stomatal conductance for the 8 to 12 most common species at each site. We also measured VC max and other comprehensive, more time-consuming physiological traits for 2 shrub species across this gradient. We focused specifically on willow species because these are 2 species which we expect to expand rapidly across our entire latitudinal gradient as part of Arctic shrubification. And then in addition to measuring all of these traits across a latitudinal gradient, we also expanded our range of environmental conditions using a nitrogen fertilization gradient. And so we measured those simple functional and physiological traits for 5 species at the long-term nitrogen fertilization experiment at Toolik Field Station, which is also on Alaska's North Slope. You can think of nitrogen fertilization as being another proxy for Arctic warming because as the Arctic warms, permafrost thaws, and when permafrost thaws, it releases nutrients that plants can then take up and use. And so we measured these 5 traits for 5 different species in 4 treatments within 4 blocks, with the treatments being a control, so no added nitrogen, plus 2 grams of nitrogen per year, plus 5 grams of nitrogen per year, and plus 10 grams of nitrogen per year. And all these treatments have been applied continuously since 2006. And just looking at this drone image of the nitrogen fertilization experiment, we can see how a nitrogen addition is simulating this process of shrub expansion, where our control plots look very similar to the surrounding tundra, but places where we've added lots of nitrogen have much denser, taller shrub vegetation. And so then we took these measurements from our latitudinal gradient and our fertilization gradient, and we paired them with spectra, and we used the spectra to predict these traits. And what I'll show now are our preliminary results. I haven't gotten around to doing too much data synthesis yet, but I think what we have so far is pretty cool. And I'll present most of my results using a standard observations versus predictions plot. where on the y-axis, I'm showing our observations of some trait or a transformed version of our trait. So in this case, the log transformation of leaf mass per area. And these observations were what we did, what we measured directly on the individuals, either in the field or in the lab. And on the x-axis, I'm showing our predictions, which come from that partially squared regression model trained only on the reflectance values of each leaf. If the model does a good job of predicting leaf mass per area from leaf spectral reflectance, we should see all of our data points fall close to the one-to-one line, which I'll be showing in gray here in a minute. And so for this model, which was predicting the log transformation of leaf mass per area from the fresh reflectance of leaves, we see that we do a really good job. Leaf reflectance strongly predicts leaf mass per area. This model had a calibration R² of 0.74, which is really quite high for any sort of biological data set. We can also color our observations by the plant functional type, or PFT, of our observations. A PFT describes the growth form that an observation came from. So whether it's evergreen or deciduous, if it's a tree, a shrub, a forb, or a graminoid. And when we color these points, we see a very clear pattern, and that's that both our observations and our predictions match exactly with what we would expect from that leaf economic spectrum. So remember, the leaf economic spectrum predicts that the short-lived, fast-growing species will have a low leaf mass per area, while slow-growing species will have a high leaf mass per area. And at the high end of both our observations and our predictions, we see plant functional types like the red points, which are evergreen trees, and the blue points, which are evergreen shrubs, typically individuals that we think of as having a very high leaf mass per area. On the other end, we see yellow and blue points, which are primarily dwarf shrubs, which we think of as having a low leaf mass per area and being in that live fast, die young end of the spectrum. We see a similar pattern for our validation data, which again was data not used to train the partially squared regression model, but we used the model to independently predict. And so we still see that our points segregate out along the leaf economic spectrum. This model had a really high R² of 0.83, and so it shows that our model predicting leaf mass per area from leaf reflectance across functional types performs really well at predicting new observations of leaf mass per area. And this could be quite useful in helping us monitor changes in LMA that arise from changes in species composition across the Arctic. But as I mentioned earlier, we aren't only interested in how leaf traits vary between species, but also within a single species. And so to start to get at that, we can look at how the partially squared regression model performs across different functional types. And so we see that the model performs better for certain types of plants than it does for others. That is, the line of best fit through the points of a given color might be closer or farther from the one-to-one line for one color than it is for another. And so, for example, you see that we do a particularly bad job at predicting leaf mass per area for the red evergreen trees, probably just because we don't have that many observations for evergreen trees in the tundra, which is primarily treeless. But we do a very good job at predicting some of these green lines, one of which is for willow shrubs. Because we did such a good job, we can build a second PLSR model that's trained only on, for example, willow data to make willow-specific predictions of LMA from leaf reflectance. This is something that a lot of other research groups in the past have struggled with, mainly because there tends to be a lot less variation within a plant functional type, or within the willow genus in this case, than there is maybe across different functional types. And PLSR models need lots of variation to make accurate predictions. When we make such a willow-specific partially squared regression model, we see that it still does a really good job. Leaf reflectance strongly predicts leaf mass per area within the Salix genus. We had a calibration R² of 0.61 and a validation R² of 0.66, making predictions across 4 different willow species: Salix alexensis, Salix pulchra, Salix histata, and Salix glauca. And so we've shown that within an expanding shrub genus, leaf reflectance can predict leaf mass per area from reflectance curves. I was surprised at how well this model did, and we did such a good job that we took things a step even further, and we asked if reflectance curves could predict leaf mass per area within a single species. And so we took the species that we had the most observations for, Salix pulchra, which is this, this light green color, and we trained a third PLSR model, this time predicting only Salix pulchra leaf mass per area from only Salix pulchra reflectance curves. And still we see that this model does a really good job. We had a calibration R squared of 0.53 and a validation R squared of 0.79. And like I said before, these sorts of species-specific trait predictions have been hard to come by. And a species-specific prediction as strong as this one is pretty novel. This success, unfortunately, doesn't come from any sort of methodological innovation that I had or something different that I did in the field, but it's mainly just a product of the fact that we were able to measure Salix pulchra leaf mass per area over such a wide variety of environmental conditions, so across both our latitudinal and our nitrogen fertilization gradients. And these environmental conditions created variation in the leaf mass per area of Salix pulchra individuals that we were then able to capture using the partial least squared regression model. Going forward, however, a model like this one will be exceptionally useful as it'll allow us to predict Salix pulchra leaf mass per area as it varies over space, over time, and in response to changing environmental conditions, allowing us to look at changes in leaf traits within a species as opposed to just across different plant functional trait types. Leaf mass per area was not the only trait that we predicted, but it was by far our most successful one. And so here we're looking at stomatal conductance, which I've abbreviated as GSW. And we see that leaf reflectance really only weakly predicts stomatal conductance. We had a calibration R squared of 0.16. And you'll notice that we have a problem of underprediction here, where our PLSR model doesn't like to predict stomatal conductance on a beta transform scale above maybe -1, whereas in the field we saw stomatal conductance getting as high as 0.5. Fortunately, we do still see, however, that our observations are segregated out along the leaf economic spectrum. So again, our red and blue points representing evergreen trees and shrubs have a low stomatal conductance because those plant functional types aren't photosynthesizing as much, whereas our yellow points, our deciduous shrubs, are much higher because they're exchanging lots of gas through their pores as they're photosynthesizing. We see similar successes and failures in our validation data for stomatal conductance. We see that similar problem of underprediction and our segregation along the leaf economic spectrum. And here we had a validation R squared of 0.24. I think it's important to note, however, that our predictions for stomatal conductance weren't a complete failure. A validation R squared of 0.24 is still a prediction, even though it's a little bit weaker than what we had for leaf mass per area. And I think that maybe our struggles to predict stomatal conductance are probably just a factor of the fact that stomatal conductance varies much more rapidly within a leaf than something like leaf mass per area. So plants can open and close their stomata, their pores, within the time frame of seconds or minutes. And so I think it's probably pretty likely that that stomatal conductance changed between when we measured it in the field and when we measured spectral reflectance afterwards. Finally, we found that leaf reflectance did not really, unfortunately, predict VC max. We had a calibration R squared of 0.05 and a validation R squared of 0.14. Remember here that we can't see any partitioning among different plant functional types because we only measured VC max for 2 different willow species, which are in the same plant functional type. And I think our struggles to predict VC max can be explained by 2 reasons. One, just the fact that we had fewer observations. So remember, each of these black points took us 45 minutes to measure in the field. And so we weren't able to get as much data. And so we didn't have as much variation to predict from our PLSR model. And then further, as I mentioned earlier, there is somewhat less of a direct link between leaf biochemistry and VC max because VC max is a physiological rate and not a physical structure itself. And so I think that also contributes to VC max being a difficult trait to predict. And so these are far from all of the results we have right now, but I think They highlight maybe something that we were very successful in doing already, something that maybe we're still working on to model conductance, and then something that maybe we can't predict within a single species, VC max. With that said, we still have a lot to do in the coming months. We're still working on measuring additional leaf traits like foliar nitrogen. Foliar nitrogen takes quite a long time to measure or extract in the laboratory, but I'm optimistic once we get these samples processed that we'll be able to predict foliar nitrogen across functional types and hopefully even within different species. Further, I'm working with a collaborator of mine, Mary Heskel, who's at Macalester College, to try to combine this data set from Alaska's North Slope with a very similar data set that she's working on from Arctic Norway. And the goal there would be to try to look at variation in leaf traits across the entire Arctic as opposed to just within Alaska. And then finally, I hope to eventually scale up these leaf level spectral predictions to the landscape level using something like the National Ecological Observatory Network's Airborne Observation Platform, which is this plane you see here. And this plane is equipped with an airborne hyperspectral instrument. And so using this imagery, I hope to maybe develop predictions of physiology and functional traits at larger scales, starting with maybe spatially explicit predictions of that nitrogen fertilization gradient, and maybe expanding up to more regions in the state of Alaska. Finally, before I let you all go, I wanted to mention a fun little side project that I worked on while we had the spectrometer in Alaska as part of the research partnership. And this side project was done in collaboration with Matt Zimmerman, who's a PhD student in fish ecology at the University of Alaska Fairbanks. Matt is currently investigating the coloration patterns in the dorsal fins of Arctic grayling, which are a fish species that live in freshwater streams on the North Slope. And this is the dorsal fin here. Matt is currently using RGB cameras to look at how different individuals have different coloration patterns in this fin. But we thought it would be really cool to use a spectrometer to try and see if there was any non-visible variation in these fins. I haven't really worked up any of this data, otherwise I would have shown it in this presentation, but I think this is a really novel application of spectroscopy. It hasn't really been applied to wildlife ecology in the past, and I think it could be really useful data in understanding how these fish select different mates or interact with one another. And so I think I'm really excited about seeing these data once I get a chance to work it out. But with that said, I'd like to extend one last big thank you to Spectral Evolution for their generosity and this incredibly productive research partnership. And I'd like to thank you all for listening. So if you have any questions, I think I can take them.